That is why it appears as a darkly stained structure. Not only that, it also has granules in it, right? Granules. And also chromosomes are tightly packed. Because of this reason also, it appears as a darkly stained organelle. Therefore, the answer would be Z, right? The answer would be D. Right. Now they're asking which structure is not found in human white blood cells. We know if we compare an animal cell to a plant cell, animal cells don't have cell walls. We know that. But over here, they have not labeled a cell wall, right? Because Z is the nucleus, W is the vacuole, X is the cytoplasm, Z is the cell membrane or the cell surface membrane. So therefore, they have not mentioned anything about the cell wall. So out from the structures given, which structure is not found in human white blood cells? You know, in any living cell, excluding the red blood cells, right? Any cells have a nucleus. So white blood cells, definitely they should have a nucleus, right? White blood cells should have a nucleus. But if you take even if you take the cytoplasm, even if you take the cell surface membrane, right? Even if you take the cytoplasm and cell surface membrane, even these are present in animal cells. But if you take the vacuole, right? The vacuole is not compulsory, right? If you take animal cells, you can't see big vacuoles in the way that you see in plant cells. Animal cells don't have big vacuoles. They have small vesicles. But if you search the internet and see, right, you can see that white blood cells do have vacuoles. But in your syllabus, we are not considering them as vacuoles. We are considering them as vesicles, right? Because there are different types of white blood cells, right? Leukocytes, basophils, right? Lymphocytes, right? All these things are there. Neutrophils, right? Because of that reason, in general, we say the white blood cells do not contain a vacuole, right? They do contain little vesicles, but not a vacuole. Therefore, W is the structure, which is the vacuole, which is not found in human white blood cells, right? If you want, you can write a review for this question. Vesicles are found in the human white blood cells, but not vacuole. Vesicles are found in human white blood cells, but not vacuole. Vesicles are found in human white blood cells, but not vacuole. Right. I'll be moving into the third one. Which molecule is a storage carbohydrate in the root cell? So root cell is actually a plant cell, right? Because inside the plant only you see the root. So root cell is typically a plant cell. So what is the storage carbohydrate in plant? It is none other than starch, right? Yes, starch is the storage carbohydrate in plant cells. Glycogen, where do you find glycogen? Glycogen is stored in fungi, right? It's the storage carbohydrate in fungi. And glycerol, it's not a carbohydrate. It's the basic components which make a triglyceraldehyde, which is a lipid, right? Glycerol makes the lipids. Glycerol and 3 fatty acids combines and it makes a lipid or a triacylglycerol. Therefore, B is also wrong. And glucose, we know it's not a storage carbohydrate. It's just a sugar that we are consuming for our daily purposes, right? Therefore, A is also not the answer. The correct answer would be D, which is starch. Now, in the B1, they have given us the actual width of the cell from P to Q. From here to here, they have given us the actual length. The actual width is 125 micrometers. But now you have given a certain paper. Actually, it's better if you have the hard copy for this, but it doesn't matter, right? Because sometimes it could vary. The value that you get will vary, but it doesn't matter. If you have got the correct answer according to the value that you have measured, you can take it as correct, right? So if you measure the paper, that means from P to Q, if you keep a ruler and measure in between P and Q, you get a length of around like 5 centimeters, right? So the paper value will be 5 centimeters. The actual width is 125 micrometers, but the value that we get is 5 centimeters, right? 5 centimeters means 50 millimeters. Now, this is the actual width, 125 micrometers, but we can't compare them. Why can't we compare them? Because they are in different units. Now we have to convert 125 micrometers into millimeters. So 125 micrometers into millimeters, you can divide it by 1000. Right? 125 divide by 1000, you get 0.125 millimeters. This is the actual length, right? This is the actual length of this blood cell, not of this blood cell, of this root cell. But the diagram that they have given measures 50 millimeters, which is equal to 5 centimeters. So we have to see what is the magnification. They have magnified it and given it to us in the paper so that we can see. Otherwise, we can't see the root cells in the plant. If we go to our surroundings, we can't notice any root cells. We just notice the roots like this, right? Yeah. So the magnified cell, the magnified cell is given. So how do you find the magnification? You need to divide the magnified length. Magnified length is 50 millimeters by the actual length, right? Actual length is 0.125 millimeters. So always the magnified length divided by the actual length, you can get the magnification. If you have done this, you will get 400 as the answer, right? You will get 400 as the answer. You don't need any units for this. Why? Millimeters cancel. It's better to put the multiplication in front. That's how they have given the 3 marks, right? That means for this multiplication also that they have given a marks. It's better. That means whenever they ask for a multiplication, you have to magnification. You have to include the multiplication sign to show it's a magnification. Right? Okay. So have you written it like that?
I didn't get like that 50 millimeter thing. I just got 0.125.
Okay. So whenever they have given us the actual width in a particular question, that means, and if they have asked the magnification, they are expecting us to measure the real life width, right? That means the width which is magnified. Then only you can find the magnification, right? Okay, so from next time onwards, if they have given a question like this, it's always better to find that's why they have given PQ, and that's why they have given a length like that. That's why they have given a line to measure that, right? So it's better from next time onwards. Now, since you have made a mistake at home after the discussion is over, redo this part, right? Like think why you didn't understand it at first. That means what was wrong, whether it was lack of, sometimes we lack concentration when doing the paper also. It's typical, like it's typical among everyone. So don't worry about that. So if you think that it's because of that reason, you can do this again and make sure that you won't do it. Then make sure that you won't repeat that mistake in the future, right? That's how we improve. Right. Now, here they have talked about yeast cells. They are talking that yeast cells can be genetically modified to produce proteins found on the outside of viruses. What do you mean by genetic modification? Genetic modification means now yeast cells in their nucleus, they already have DNA. But you are taking this DNA out and you're adding another piece of extra DNA, right, from another organism. And this particular gene, right, this particular gene taken from that other organism can actually produce a good protein. For example, let's assume there is an animal which is found in the Himalayan mountains, right? There's no such animal like that. Just assume there is an animal which is found in the Himalayan mountains. Their cells produce a protein that can kill cancer cells, right? Produces a protein that can kill cancer cells, right? Now, the scientists get to know about this animal. So scientists travel to Himalayan mountains and they kill these animals, right? Then the whole species is going to get extinct. If they are going to kill the animals and if they are going to take the cells so that they can take the protein and cure the cancer cells cure the cancers in humans, the animal species is going to get extinct because that's a rare animal species as well. So what can we do? We want to protect the animal species as well as we want to take the protein from them. That is why this genetic modification is very important, because we can take just one cell from them and we can insert their DNA into the yeast cells or into another cell which can rapidly multiply. So these yeast cells will rapidly multiply and they will produce billions of cells, which produces that protein. We just have to take that protein from those yeast cells and give it to the humans, right? So rather than killing those animals, we are doing a good thing because we are using genetic modification to produce that particular protein that we need. That's how nowadays vitamins are produced. That's why vitamins are available at a commercial, that means available at an affordable price these days, because they're produced using genetic modification. Right. Now, you can see yeast is a single-celled organism. You have genetic modification in your syllabus. That's why I mentioned a bit about that. So yeast is a single-celled organism, and yeast has a nucleus. It has a cell wall made up of chitin. Which group of organism does the yeast belong to? You know, if their cell wall is made up of chitin, they are not plants. And you know, animals don't have cell walls. And because they have a nucleus, they can't be protoctist as well. Therefore, they are going to be fungi, right? Because fungi, they have specialized features. For example, they have a cell wall made up of chitin, and also they store glycogen as their storage carbohydrate, right? So the answer would be B. Let's write down a review for that. In fungi, the cell wall is made up of chitin. In fungi, the cell wall is made up of chitin, and the storage carbohydrate is glycogen. In fungi, the cell wall is made up of chitin, and the storage carbohydrate is glycogen. The storage carbohydrate is glycogen. Right. Now, in the second question, give a reason why viruses are not considered to be living organisms. Right. Now, if you take animals, if you take fungi, plants, prototists, all these are considered as living organisms, but not viruses. That is because they do not have a cellular structure, right? They do not have a cellular structure. That means the typical things which is present in a cell, in a typical cell is not present in these viruses, right? And also they don't even have a proper nucleus either. They just have a piece of RNA or DNA in them, right? Every organism, right? Every living organisms have DNA as their genetic material. But if you take the viruses, they don't have DNA. It could be DNA or RNA. In some viruses, DNA is the genetic material. In some viruses, RNA is the genetic material. So they're asking why we are not considering viruses to be living organism. One main reason is they can't reproduce on their own. You know the characteristics of living organisms. There are several characteristics: growth and coordination, right? And also, what else? Reproduction, evolution, right? These are the characteristics of living organisms. But viruses can't reproduce on their own. They need a host cell to reproduce. That could be considered as a reason to why that viruses are not considered as living organisms. So viruses do not reproduce independently. If you have written that answer, you can take 1 mark. And
I've written that
yes.
I've written that viruses do not carry out any life processes except reproduction. Would that count?
Yeah, like viruses do not move, viruses do not grow, viruses do not respire, viruses do not yes, that is correct. Viruses do not carry out life processes. It's correct. Yes, that could be considered as a correct answer because that is given in the marking scheme, right? Yes, no worries about that. But the reason is, now you have right that they do not reproduce. In brackets, it's better if you could write independently, right? In brackets, but your answer is correct. No worries. Your answer is correct, but to be on the safe side always, better to write next time, better to write that they do not reproduce independently because they reproduce, right? To mention that they do not, that means to tell the examiner, to show the examiner that you know that, it's better to write the word independently as well, right? Otherwise your answer is perfect because it's given in the marking scheme under the additional guidance, right?
Yeah.
Okay, now B part, a species of yeast is genetically modified to produce a protein found on the outside of hepatitis B virus. Okay, so here they have taken a gene from the hepatitis B virus and they have included it into the yeast, right? They have attached it into the yeast genome. In the yeast genome, they have included this blue hepatitis virus gene, right? So that this yeast can now be pathogenic. That means this yeast can now produce that protein which this hepatitis B virus is producing. So now you can use these yeast cells to make the vaccine. Because in vaccines, you are basically using the same organism which causes the disease. But the only difference is they are not strong viruses. That means they are not the strong version of that virus. They are weakened and they are attacked that means they are weakened and they are inserted into that particular vaccine. So that we don't get that actual symptoms, but we do get part of the symptoms, and our body produces the required antibodies as well as the secondary memory cells. Because after we get that pathogenic DNA inside especially the viral DNA inside our body, what happens is our body's immune systems recognize this viral DNA and they start creating the T lymphocytes, the B. Yes?
Nothing, miss.
The T lymphocytes, the B lymphocytes, they start creating secondary memory cells, right? So these secondary memory cells can stay within our body for a long time. And in case if we get administered, that means if we get in touch with that particular virus later in our life, what happens is these secondary memory cells will quickly recognize, okay, these were the cells that entered this particular person before. Now I can kill these cells easily by producing those antibodies, right? So basically, this protein is used to make vaccines to prevent people being infected with hepatitis B. Okay, the gene for this protein is inserted into the plasmid. The plasmid is then used to modify the yeast cells. Okay, even though now you might have heard that plasmids are only seen in prokaryotes, but yeast cells also have these plasmids. Yeast cells also have some plasmids, right? So basically, in the hepatitis B virus, there is a DNA. You take that particular gene, you take that particular gene which codes for a particular protein. That means which codes for the specific protein that creates its pathogenicity. And you take that piece of DNA. And in these plasmids are also yeast DNA. So you take this yeast plasmid out, right? Yeast plasmid out. I'll draw it in blue so it's easy to identify it, right? You take the yeast plasmid out, and we don't have scissors inside our cells. We need to cut this DNA. So the scissors inside our cells are called as DNA ligase. Oh, no need DNA, you can call it ligase. They are the scissors, sorry, not scissors, they are the glues. They are the glues. Restriction enzymes are the scissors which are used to cut any DNA. We don't have any scissors inside our cells. So to cut any DNA at a specific site, you use restriction endonuclease or restriction enzyme. So you take the plasmid. Now you need to cut this plasmid. Why? Because you need to insert this piece of DNA. So you use the restriction endonuclease, which is a scissor, and you cut the plasmid like this. See? Now I have cut the plasmid. The other part of the plasmid is over here. Now I take this DNA and I keep this part of the DNA gene over here. Now there is an issue. I have to paste this DNA. We don't have glue or we don't have gum inside our cells, not even in the laboratories. So we have to use an enzyme which acts as a glue. The enzyme which acts as a glue is the ligase. So we take the ligase and we paste. We paste this piece of DNA, this segment of new DNA into this plasmid, and we reinsert this plasmid into our yeast cells. So our new plasmid will look like this now. And now, yes?
Miss, I kind of wrote like restriction enzyme is used to cut the desired gene, and DNA ligase is used to paste it onto the plasmid.
DNA ligase is used to place it into the?
Is used to paste it onto the plasmid.
Onto the plasmid. Yeah, it's correct. DNA ligase is used to paste it, right? It's correct. No issue. Attach, paste. You can use those words, right? Okay, so I hope you understood it. You can put it back into the back into the yeast cell, and they will reproduce. And when they reproduce, they create that same gene. They create the same plasmid inside them now. So all those yeast cells can now produce that particular protein which hepatitis B virus used to produce. So that is the story behind this. So the two enzymes that you use to produce plasmids containing the gene for hepatitis B protein is the restriction enzyme, or you can use restriction endonucleases, but better not to write endonucleases only, because even though they have given marks in this particular marking scheme for endonucleases, the correct word is restriction endonucleases. The full form of endonucleases is restriction endonucleases. So it's better not to write endonucleases alone. It's better to write restriction enzymes or restriction endonucleases to cut the DNA or to cut the gene or to cut the plasmid or to remove the gene. 1 point for that. And ligase joins, paste, attaches the DNA to the plasmid. 1 point for that. So already 2 points for that particular question. Okay, so this is a picture of the virus that I have taken. You can see DNA or RNA present. You can see there's no nucleus. The typical cellular structure is absent in the virus, right? Because there's no cytoplasm, there's no cell membrane, right? It's just an envelope.
Isn't this a fungi?
This particular one?
A yeast. I think it's a yeast or fungi cell.
No, it's a virus. Even though I pasted it over here, it came to this part, this virus. I wanted to show this over here, virus. Because fungi are eukaryotes. Fungi only contain DNA as their genetic material. They are eukaryotes. Even eukaryotes, prokaryotes, they contain only DNA as their genetic material. Only viruses contains DNA or RNA, right?
Yeah, I confused that with the structure of a bacteria.
Okay, I pasted it because I did not have space over here. Right. Now the C1, here they have asked, the diagram shows an industrial fermenter. You could see that in the first paper that we discussed also, they have asked about this fermenter. So fermenter is quite popular, right? Okay, so you can see, explain the function of the temperature monitor and the cooling jacket. So this is the temperature monitor and this is the cooling water, right? So what happens is in the fermenter, fermentation takes place. So fermentation actually releases heat energy. Fermentation releases energy. This energy could be in the form of heat. So when excess heat energy is produced, what happens to the temperature inside the fermenter? The temperature inside the fermenter increases beyond the optimum value. Optimum means sort of like the maximum, right? But it's not really the maximum. Optimum is the required temperature for the enzymes to work, or the temperature that the enzyme works best at. So if the temperature increases beyond this optimum temperature, the enzymes will actually denature, because you use enzymes, you use different microorganisms in this industrial fermentation. So they could get denatured because of these high temperatures. In order to prevent this, there is a temperature monitor which will always detect the temperature. So in case if the temperature is beyond a certain value, if the temperature is above a certain value, it quickly opens this valve or opens this tap. Sorry, it's not a tap. This is the tap. This valve over here. It opens the valve so that cooling water will enter into this cooling water jacket. Afterwards, due to this cooling water, again this fermenter will go back into its normal temperature. So that is why the temperature monitor and cooling water is there. Since they have used the word explain, you have to explain the whole process. That means you have to mention all the reasons why temperature monitor and cooling jacket is there. So it's better to include, even though they have given 3 points, it's better to include all these things. But you get, if you have written any of the 3 points which I have mentioned over here, you get the 3 marks. So respiration or fermentation is the process which will release the heat. So what happens then? The temperature monitor will check the temperature and it will open the valve to let the cooling water in. For that you get 1 mark. And then when you let the cooling water in, it will reduce the heat because the cooling water can absorb the heat, right? And then it gets out of the cooling water jacket. And why do we do this? We do this to maintain an optimum temperature. What is the reason why we need to maintain an optimum temperature? To stop the enzymes from denaturing. If the temperature exceeds above the optimum temperature, enzymes are proteins. Proteins get denatured, their shape changes. So to prevent this from happening only, we have to maintain the temperature at the optimum temperature. If you have written any of the 3 points, you can get the 3 marks. Right. Now, in the second one, they have asked, explain why air is needed in the fermenter. So some air, but in this fermenter, they require air. The reason is because they need oxygen to carry out aerobic respiration. Your first point should be providing oxygen, because air consists of nitrogen, oxygen, right? A lot of things. So you must mention to provide oxygen. For that only you get 1 mark. And why do you need oxygen? To make sure that the yeast will carry out aerobic respiration. Otherwise, yeast can carry out anaerobic respiration and there would be different products. Ethanol will be produced. We don't want that result to take place, right? By anaerobic respiration of yeast, ethanol could be produced. In order to prevent anaerobic respiration only, we need to make sure that oxygen is given to that fermenter, right? They have not given us what the fermentation process is, right? But by telling that air is needed, we should understand that this yeast is carrying out, that the yeast is required to carry out aerobic respiration. Some fermenters might need the yeast to carry out anaerobic respiration in order to produce ethanol. Why? To produce alcoholic drinks, right? Sometimes, maybe if depends on the purpose, right? For some purposes, they might let the air in. For some purposes, they might not let the air in to that particular fermenter. So if you have written the 2 points, you can get 2 marks. Then they're asking, explain why the air is filtered before going to the fermenter. You know, air can carry a lot of microbes. So definitely it needs to get sterilized first, or it needs to get filtered. So what is the reason? To prevent other microbes. Now, apart from writing microbes, if you have written bacteria or fungi or pathogens, you can get that particular 1 mark, right? To prevent other microbes, bacteria, fungi, pathogens, you can get that 1 mark, right? And also, you must mention to get the other mark, why do you prevent the entering of microbes? To make sure that the product will not get contaminated, right? So you can write down, in order to ensure that the product will not get contaminated. For the word contamination, you get the other mark, right? For the word contamination, you get the other mark. So if you have written, the word contamination must be there because they have underlined, because they have not given marks for keeping it clean or writing germs. Don't ever write germs. Be specific. Use scientific words like microbes, bacteria, fungi, pathogen, right? Don't write germs. In Edexcel IGCSE biology, don't write germs because they are not giving marks for that, right? Now, the diagram shows part of the food web forming an ecosystem in a region of North America. Which of these organisms in the food web is a primary consumer? Right. So, primary consumers are the ones which depend on the producers. Producers are the ones which uses sunlight to make their own food. So, the producers are grass and aspen trees. Then who are the primary consumers? You can see there are 3 primary consumers. Mouse is a primary consumer, beaver and elk. These are the 3 primary consumers. Therefore, out from these 3, they have only given beaver over here. From these 4 answers, only beaver is the primary consumer. So, answer would be A. Right. In the second question, they're asking, let me know if I'm too fast. If you want me to stop anywhere, you are free to stop me, right? Okay. Second one, they are asking, what is meant by the term ecosystem? So, ecosystem, now you know, first of all, there is an individual. So, all these individuals get together to make a population. And different populations, right? Different populations makes a community. And community with the environment, that means community means the living things. Different populations of different species, lions, zebras, right? Giraffes, like the Madagascar, you have different populations. So there is a community. So after that, the community and the non-living environment, the rivers, the grass, the sunlight, all these things, water, all these things make an ecosystem. So basically, you can easily write, if they ask what is meant by the term ecosystem, if you have mentioned that it's a combination of the community and the environment, you get 1 mark. Or else you can write it down like this. If you have mentioned biotic and abiotic factors, biotic and abiotic factors, biotic and abiotic parts together, that also means it's an ecosystem. Or else you can write it in the normal terms, like all living things and all non-living things. But the word all is important because ecosystem means everything, right? All the non-living things as well as all the living things, right? And also, if you have written if you have just if you have written all organisms and the environment, instead of writing community, if you have written all organisms and the environment also, you can get that particular mark. Right. Yes.
No, miss, I got the mark. Like I wrote, an ecosystem is the combination of biotic and abiotic factors.
Yes, that is correct. Right. Now, B1, they are saying that wolves were hunted in the region of North America until they became extinct in 1926. Right. So they are saying that the extinction of wolves damaged the ecosystem in these ways. Population of mice, hawks, beavers had decreased, and the population of producer species, that means the grass and the aspen trees, also have decreased. And also, they have mentioned that beavers are important because they cut down the old trees, and also they cut down the old trees, and also they create shelters that the organism nest in. They're asking discuss why the extinction of wolves damaged the ecosystem. When they say the word discuss, right, you don't have to explain anything. You don't have to give the reasons for anything. You just have to make sure you are writing why the extinction of wolves have damaged the ecosystem. You are only answering the question. You're not giving reasonings, right? You're just, by looking at the diagram, you're saying because wolves have got extinct, elks and coyotes have increased, right? Because they don't have any predators now, because wolves have become extinct. So elks and coyotes are now becoming increased. Or else you can write down fewer elk and fewer coyotes were consumed, because now no wolves are there. So for that you can get 1 point. Right. Now fewer elks and fewer coyotes were increased, means when elks are increased, they are eating the producers, right? Aspen trees, grass, both are their food. That means both are their food sources. Therefore, the grass and the aspen trees would decrease. So you can write down more consumption of plants. For that, you can get 1 mark. And also, when the coyotes increases, the number of mice and the number of beavers would decrease. Why? When there is more coyotes, that means there are more predators for the mice and the beavers. Therefore, mice and the beavers will be eaten. More mice and more beavers will be eaten. Right? So for that, you get 1 mark. And also, because the elks are eating the grass and the aspen trees, there will be little amount of food for the mice and the beaver. You can look at this diagram and you can see everything that you see. So there will be less food for mice and less food for the beavers, right? So when you are writing this, you already have 4 points over here itself. Not just that, when the mice is being eaten by the coyotes, there will be less mice for the hawk, right? There will be less mice for the hawk. Therefore, the hawk, you can hear me, right?
Yes, miss.
Because I'm getting a call to my other phone. I'm wondering whether the hotspot, I'm using hotspot, that's why, right? I'm not answering the call. Okay, so you can see the hawks, right? The mice is being eaten. Because of that, the hawks will not have enough food. So hawks will also reduce. Just because one species is being removed, there are a lot of changes happening in this ecosystem. So less food for the hawk. Therefore, the number of hawks, that's why see the population of hawks are also being decreased because of that. And also, now less beavers are there. Why are there less beavers? You don't have to explain why there are less beavers, right? You know why there are less beavers. Why? Because less aspen trees, and also more coyotes are there. So therefore, why? Therefore, the number of beavers are less. As a result, what happened? All trees will not be removed. They're asking the damages to the ecosystem. That means not just the living things, also the non-living things, but trees are living things, right? So you can mention about the trees as well. All trees are not removed. So when all trees are not removed, what happened? All trees are not removed means they are going to shade. That means they are going to shade the younger plants. Therefore, less penetration of sunlight to the growing plants. Therefore, photosynthesis will not properly happen to these younger plants, younger trees. So younger trees are shaded, therefore they will have less photosynthesis. Not just that, when these little plants are eaten, when the aspen trees are being eaten heavily, right, there will be loss of plants in that ecosystem. Due to the loss of plants, when plants are being removed or when plants are being cut down, there will be soil erosion. There will be soil erosion. Now that is an abiotic factor because they're asking about the ecosystem. You can mention about the soil erosion as well over here. So soil erosion will happen due to the loss of plants. And not just that, because these because beavers cut down the old trees, and beavers create shelters for the other organisms. Because beavers are not there anymore, the old trees are not cut down. As a result, there will be fewer shelters for the organisms. So you just have to look at the diagram and write what you see. If you have written any of the points, you get 4 marks. If you have written any of the 4 points, you can get 4 marks, right? You can write down like this as well. Fewer elks or fewer coyotes are hunted, or fewer trees, fewer small plants are there, or but don't write no or fewer beavers, because they have not given marks for no or fewer beavers. No or fewer beavers, they have not given marks for that particular answer. So now at the exam, you don't know what to write and what not to write, right? That means you don't know whether they would give marks for no or fewer beavers. They have given marks for mice part, but they have not given marks for the beavers part. So at that time, you have to write everything that you see, because they have given a big space for you to write everything, right? Because they are not just expecting you to write the marking scheme points. Those marking scheme points are not for the students to look at. Those are for the examiners to look at and mark your answers, right?
Yeah.
So you have to write a big description about this. And if you have written those points, you get the marks, right? Okay. Now let's move on to the C part. In 1995, 14 wolves were introduced into this region. The diagram shows the change in wolf population. Calculate the percentage change in the wolf population between 1996 and 2004. So in 1996, the wolf population is 50. In 2004, the wolf population is around like 180. Because this is 180, this is 190, this is 200. Because every single unit like this, it includes 5. That means increase of 5, 150, 155, 160, 165, 170, 175, 180. Likewise, increase of 5. So therefore, it will be 180. They're asking the increase, percentage change. So the change should be final value subtracted the initial value, which is 180 subtracted 50, 130. And how do you find the percentage change? The change divided by the initial value. What was the initial value? 50.
50?
Yes, initial value is 50. Multiplied by 100, you get 260%, right? You get 260%. If you have written just 260% without writing any workings, you can get the 2 marks. But it's always better to write the workings in the space that they have provided. So 260%. Right. Then they have asked, suggest why the wolf population changed between 2004 and 2012. So we don't know any reason. Suggest means we have to think and write. We have to think about the different ways that the wolf population might have decreased from 2004 to 2012. You can see there is a big decrease. There is a deep decrease, right? So we have to think and write down the answers. So in case if they ask any question like this, these are the answers that you can think of. It's better to keep these answers in your head, because at the examination, sometimes due to stress and everything, you might not be able to think about new answers. It's better to remember these answers, not memorizing, but just remember, right? So the wolf population might have decreased because their food might have run out. Because due to a disease or something, all their prey have been killed. As a result, their food might have ran out. Because of that, the wolf population have decreased. So that could be one particular answer. And not just that, one particular wolf might have got a disease, and that disease might have spread throughout that population. As a result, that they might have got decreased, and only the adapted ones have been able to survive from that disease. And also, hunting might have increased during that period. Don't just write hunting. Better to write hunting might have increased from 2006 to 2012 as a reason, from 2004 to 2012 as a reason they have decreased. And also, new competitors might be there. When predators have competitors, when other predators come and when they depend on the prey, what happens is the number of food that they have decreases. So the number of individuals in that population, in that species, will also decrease because they don't have enough food. And also sometimes it might be because they have lost their habitat, or due to a change in the temperature or something, due to change in environmental factors, they might have migrated. Those might be the reasons. So if you have written any sensible answer, you can get 2 marks for that. Then they have said the reintroduction of wolves caused changes in the population of some plant species. Describe how the population size of a plant species in this region could be determined. Right. So in order to find the number of plant species, the typical way of determining the number of plant species is by using a quadrant. And since they're asking to describe it, we have to describe the way that we are using the quadrants. So when we use quadrants, we can't place all the quadrants, that means closer to each other. We just have to randomly place. We can't decide, okay, here I'm going to place one, here I'm going to place one. We can't place them in a particular pattern. We should place them randomly. So then only we get a accurate answer or a more reliable answer. So you should randomly place them. If you have mentioned using quadrants and placing them randomly, you don't have to write them in the same sentence structure. You can write it down in your own words, place them randomly, right? After placing them, you just have to count the number of plants in each quadrant. And then you have to calculate the mean, right? And then by taking that particular value, that means you take that particular value for a small scale, right? For example, for 2 meters squared only, you calculate the number of plants. But the whole area could be around like 100 meters squared. So you just have to multiply it by 50 to get the accurate answer for that area. So you scale up the whole area. So if you have given any idea that by calculating it for a particular value, you can find the real value, something like that, you can get that particular point, right? So using quadrants, in many papers, they ask this quadrant part. So we should know how we should actually use a quadrant. Instead of writing a quadrant, if you have written grid area, that could also be considered as a correct one. But if you have written quadrant, right, wrong spellings like this, right, you might not get that mark. You can't write quadrant, you should write quadrant, right? They're considering the marks as well. And if you have written quadrant, they're considering the marks. If you have written quadrats, instead of writing quadrats, that means plural, you get 2 marks. Quadrats, that means by using the plural form of quadrats, you are implying that you are using many quadrats to calculate the number of plants, right? So you get 2 marks. Yeah. But we can't always depend on the plural of that means every time we can't say in all the marking schemes, just because we write the plural form of quadrats will get 2 marks. So better to, if they say describe, better to explain how we are using the quadrats to get a more reliable value for the number of individuals which are living in a particular area, right? So this could be considered as a typical answer that you can repeatedly write for any question that they would ask, in case if they are asked to describe how we are using quadrats. But here they have not mentioned the word quadrats. You have to think and write down the word quadrant, right? Okay. Now,
I have a question.
Yes.
What is the difference between a quadrat and a quadrant?
A quadrant means it's used in mathematics, right? Quadrant. The word something like this, right? Quadrant. I think it's used in mathematics. It's not used in science. Let me check it. Let me check it. Quadrant. Yeah. Yeah, a quadrant is 1/4 of a circle. It's 1/4 of a circle. That's what you call as a quadrant. Yes, it's used in mathematics as well. Those quadrant equations, I remember like this. Yeah, not in science. Yeah. I cannot remember mathematics that well because I did it a long time ago. But I remember that there was something like that in mathematics, right? You can see in the fourth one, the diagram shows part of a human thorax with structures labeled R and S. Okay, so they're asking what is the structure, what is the name of R? So structure R, you know, this is the bronchioles, right? Sorry, bronchus, the right bronchus, the right bronchi, and the left bronchi. The singular word is bronchus. And these tiny branches are the bronchioles. The tiny branches are the bronchioles, and at the end, you can see sacs like this. These are actually, they have only single, that means they have only a what do you call, a single membrane. That means one cell thick membrane is there to allow gaseous exchange, and they're coated by a lubricant as well. The reason is, in case, that means they would collapse. That means to increase the surface tension, otherwise they would collapse, because they create, that means they create a large surface area for the gaseous exchange, and since they are air filled sacs like this, they could easily collapse when these lungs are being, that means when these lungs are being getting expanded, and when they are being relaxed, they could get, due to their high surface tension, they could collapse. Because of that, they have a lubricant in order to prevent them from getting collapsing. So the surface tension is actually being discussed in physics. If you understand the surface, you don't need to understand the surface tension part, but because of the high surface tension, there is a tendency that they could get collapsed. Because to prevent them from collapsing only, you have the lubricant, and also they are moist. Why are they moist? To allow the gases to dissolve. For example, oxygen and carbon dioxide, they can dissolve in this moist environment. This facilitates the diffusion between the alveoli and the blood capillaries. Alveolus is also one cell thick, blood capillaries is also one cell thick, and the alveolus is moist, so the gases get easily diffused, and oxygen dissolved, and easily oxygen can diffuse into the blood capillaries, and carbon dioxide can diffuse out from the blood capillaries back into the alveoli. So those are the special features of alveoli. And this is the trachea. You know, the trachea has rings of cartilage in it as well. So you can see the diagram that they have, the part that they have shown is the right bronchi. Sorry, when you see the human form in this shape, even though this seems like the right section, it's actually the left side, right? I hope you know that, because this is the left side, right? This is the left bronchiole, this is the right bronchiole, so this is the bronchus. Right, now they're asking, describe how changes in structure S, which is the diaphragm, enables a person to breathe in. So when we are breathing in, you must mention what happens to structure S. Since they're asking us to explain it, they're looking for reasonings. So diaphragm, when we are breathing in, what happens to the diaphragm? Diaphragm will contract. Diaphragm will contract. When our diaphragm contracts, it increases the volume. When our diaphragm contracts like this, it increases the volume. It increases the volume in our thoracic region. So it increases the volume. That is one answer. Do you know in physics, we have this PV is equal to K, right? PV is equal to K. It's the Charles law, I think. Yeah, Charles law. PV is equal to constant. When pressure increases, volume decreases, right? When pressure increases, volume decreases. So when you increase the volume, the pressure decreases. you know outside pressure is high now, inside pressure is low. So from a high pressure into a low pressure only everything flows. That's how everything happens, right? The tendency is always from a high region to a low region. For something to flow, it should be in a high region, then only it can flow easily to a low region without using any energy. So what happens during breathing in is from a high pressure region now to a low pressure region, blood will flow in. Why? Because when you increase the volume, the pressure inside will decrease. So the answer should contain diaphragm contracting, increasing the volume. As a result, the blood pressure will decrease. Or you can make sure you can write down the air flows down a pressure gradient. Why? From a high pressure region to a low pressure region, it's a pressure gradient. It doesn't go against a pressure gradient. It flows down a pressure gradient. So you can mention that word in as well. Or else instead of writing diaphragm getting contract, you can write down that diaphragm has a less dome shape, right? That means when contracting, what happens is this diaphragm moves down like this. So you can see the dome shape is reduced now. The dome shape is reduced. As a result, there is more volume as well. Now, before we had this much of volume. Now, when the diaphragm has contracted, we have this much of volume. So the volume has increased actually. So the pressure decreased. So that is the complete answer. And I have taken some other information as well, which is in the syllabus. You can see that there are, that means apart from the diaphragm, we do have two other muscles called the external intercostal muscles and the internal intercostal muscles. You must know that they also work antagonistically. That means when one contracts, the other relaxes, right? During breathing in. So you should know about that part as well. Now, let's move on to the B1. Here they have said a scientist used this method to investigate the effect of exercise on the ventilation rate of a person. Okay. They are asking us to find the independent variable in the investigation. They have given some steps to do the experiment, and finally they are calculating the ventilation rate in decimeter cube per minute, right? So here they are calculating the independent variable. Here they are asking us to state the independent variable. Independent variable is the variable which does not depend on any other value. For example, it is the one that you are actually determining, that we are setting, and all the other variables depends on it. So what is the value that we are determining? We are the ones which are determining the cycling speed. If we want, we can increase the cycling speed from 0 to 10. From 0, then 10 to 30. Rather than going it within increase of 5, in increments of 5, we can go with increments of 10 if we like. Right? So it's the one which we are determining. Therefore, the cycling speed is the dependent variable in this investigation. Right. So, since you have you done this part?
Yes, miss.
All right. Okay. I thought of reading this part. Otherwise, it would be sort of incomplete. But it's okay. I'm not going to read this part. Do you want me to read this part?
No, miss. It's okay. I did the question.
Okay. So you can see in the second one, at a cycling speed of 35 kilometer per hour, that means 35 kilometer per hour, he has a breathing rate of 25 breaths per minute. Okay. They also have a ventilation of 65 decimeter cube per minute. Okay. Then they are asking, calculate the volume of air in centimeter cube in each breath. That means ventilation rate means the amount of air they take in per minute, not per breath. This is the amount of air they take in per breath. This is the amount of air. Ventilation rate means the amount of air that they take in. See, they have said ventilation rate per minute. So the amount of air or the rate of amount of, that means the amount of air that they take per minute. That is what you call as ventilation rate. So basically, that rate will be 65 decimeter cube. 65 decimeter cube, let's convert it into centimeter cube because at the end we have to find the volume of air in each breath in centimeter cube. So first I'll convert 65 decimeter cube to 65 centimeter cube, which will be 65,000 centimeter cube. Why? 1 decimeter cube equal to 1000 centimeter cube. Now I have found the amount of air that that person is breathing in per minute. Per minute, not per each breath, per minute. Right. Here they have said the number of breaths that the person is taking per minute. The number of breaths per minute will be 25 breaths per minute. So 65,000 of air is taken in for 25 breaths because both are for minutes, right? 25 breaths per minute and 65,000 centimeter cube per minute of centimeter cube per minute. Centimeter cube of air is taken in per minute. Did you understand that part? 65,000 centimeter. Yeah, 65,000 centimeter cube of air is taken in per minute for 25 breaths of air, which is taken in per minute. So therefore, we have to divide 65,000 by 25 breaths because this is also for minute, this is also for minute. So minute and minute cancels off. So this will be the amount of air, this will be the number of breaths. So we can, after we divide 65,000 by 25, we can find the volume of air that they are taking in for each breath. So 65,000 divided by 25 will be 2,600 cubic centimeters. If you have directly written the answers, you get the 2 marks without writing down any workings. But it's better to show the workings so that you also know that you are correct. Then you know the third one. Comment on the effect of increasing cycling speed on the ventilation rate. So you can see when you as you increase the cycling speed, the ventilation rate also increases. So here they're asking us to comment, and they are also asking us to refer to the data in the table. So you can see the ventilation rate increases. Ventilation rate will increase. What does that mean? When ventilation rate increases, we are taking in more oxygen to our body because the number of air which we are taking in is increasing. As a result, the amount of oxygen that we are taking in also increases. When the amount of oxygen that we're taking in increases, aerobic respiration. Aerobic respiration is the process of producing energy in our body. Aerobic respiration requires oxygen. Therefore, when we take in more oxygen, what happens? Our respiration rates also will increase. As a result, it will release more energy, right? It will release more energy. And not just that, when we are increasing our cycling speed also, we consume a lot of energy. So as we consume a lot of energy, what happens? We require more oxygen. We require more oxygen. So more oxygen will contribute to more respiration in turn. And when we are increasing our cycling speed, that means faster muscle contractions will be there. So for faster muscle contractions, we again need more air. We need more respiration. That means we need a faster rate of respiration. For that, we need more air. Therefore, volume of air breathed per breath also increases as our speed increases. So here it's like a loop. You just have to explain the same thing over and over again. That means because when ventilation rate increases, it takes in more energy. You have to first you have to mention what happens to the ventilation rate. It increases. You know that. Why does it increases? Because as cycling speed increases, when cycling speed increases, our rate of aerobic respiration increases. You can mention that if you like. That means you have to write your answer. Rate of aerobic respiration increases. When aerobic respiration increases, it requires more oxygen, right? So if you write more oxygen, you get another point. You might not get point for the aerobic respiration, but because you have explained it that way, you are leading to the next point. It takes in more oxygen. Then why do we need, because we need more oxygen, what happens to our ventilation rate? It increases further, right? Yeah, so it increases further. And also you can then mention when we increase our cycling speed, further muscle contractions takes place, faster muscle contractions takes place. As a result also, we need more energy, right? We need more air. That means volume of air breathed in per breath also increases. So it's like a loop, same thing is repeated. These sort of questions are tricky, but if you can write, that means you need a bit of a practice in these questions. That means range of past papers need to be practiced in attempting these sort of questions by looking at the table in order to explain. From this one, you can understand the pattern, right? So that means you will only get 1 or 2 questions like this in your paper. All the other questions are easy to be, all the other marks in the paper is easy to be taken. But these sort of ones are sort of like tricky. You might get 2 out of 4, but doesn't matter. You still can go for an A star, right? You can still go for the highest
because I got 2 out of 4 for this question.
Yeah, it's okay. It doesn't matter. Don't be demotivated by looking at this part. There are so many other areas that you can get the marks, right? Okay, but try your best to go for 4. You just have to identify the pattern. It's okay. It's a loop, right? If you can just explain something at the end by looking at this table, because this is not a theory based question. This is just by looking at the table, you have to get the answer, right?
Yeah.
Okay. So now here they have said, state how the scientist could make the investigation more reliable. So basically, like in most of the question, when they're asking how do you make it more accurate, how do you make it more reliable, the typical answer that anyone writes is by calculating the average or calculating the mean. Or else, if you don't want to write like that, you can say that you can use more people, right? Sometimes here they have used only 1 person, right? But rather than using 1 person for all the things, you can use more people, and that means you can take a variety of results rather than using 1 person. So instead of writing calculating the average, you can use more cyclists, more people, right? For any of those things, you get 1 mark. Or else you can go with the typical answer, calculating the average. Here they have shown us a photograph shows a variety of chicken called the silky chicken. Okay, so silky chicken have feathers that have a fluffy appearance. Okay, feather structure is controlled by a single gene, which means it's monogenic. That means, sometimes some structures, some of our features are monogenic, which means they're controlled by only 1 gene. But some of our features, they could be polygenic. That means they are controlled by more than 1 gene, right? But monogenic ones also can have different alleles. For example, if we take our eye color, right? It could be blue, it could be brown, it could be black, it could be green, right? It could be several colors. These are alleles. These are not genes. The gene for eye color, it's only 1 gene is there. Only 1 gene is there for eye color. It's located in a particular chromosome, but there are different varieties of that gene. Blue gene is there. Sorry, blue variety of that gene is there. Brown variety is there. Black variety is there. Green variety is there. So that is monogenic, but it has different alleles. Because it has different alleles, you can call it polyallelism, which is not in your syllabus, right? You only have monogenic and polygenic, right? Polyallelism is not being discussed in a thorough manner, so you don't have to worry about that, right? Having different alleles means it's polyallelism. But sometimes it could be monogenic, that means only 1 gene, but it has only 2 genes, 2 alleles. 2 alleles, like see this allele, normal feather allele and the silky feather allele. It's the allele is for the feather structure. The gene is the feather structure. So only 1 gene is there, monogenic. Only 1 gene, feather structure gene, right? And it has 2 alleles, which is silky gene and the normal gene, right? No, sorry, it has 2 alleles, which is the silky allele and the normal allele. but only one gene, monogenic feather structure is the gene, right? So those sort of genes which have only 2 alleles, you don't call that polyallelism. It's a normal one, right? Almost all the genes typically have 2 alleles, but the ones which have more than 2 alleles are what you call as polyallelism, but it's not been taught, that it's not been discussed thoroughly in your syllabus, right? You only have to know about monogenic and polygenic. So polygenic means what? Some of our features are controlled by more than one gene. So there are several chromosomes in our body. These chromosomes have several genes in several parts of them, right? So if I take the first chromosome, if I take the second chromosome, if I take the third chromosome, there could be genes in them. Or sometimes one or more genes can coordinate for some of our features. For example, our skin color, right? Our skin color is actually polygenic. It's not controlled by only one gene. It's controlled by many of the genes. And our height is also controlled by, you don't have to know what these things are, right? You don't know what that means. Just remember the polygenic examples, it's okay. But no need to remember the monogenic examples. They usually give it like height, skin color, and also intelligence. These are polygenic ones, right? But there are some genes, they are monogenic. You don't have to really know about monogenic ones, but it's better to have an idea about the polygenic ones, right? Now we have, they're asking what is meant by a gene. Okay, so you know, there is a molecule called DNA. It's a double helix molecule. We discussed it on Tuesday. It's a double helix molecule. You know, when this double helix molecule get associated with the histone proteins, right? It's called a chromosome. It's called a chromosome. So if you take, now you know what is a DNA, what is a chromosome. When DNA is associated with proteins only, you call it a chromosome. So what is a gene? A segment of the DNA which codes for a protein. You know, this DNA has several parts. Some of the parts are not coding for any DNA. Last time I told you they are called as exons, which you don't need to know, right? You only need to know that there are parts in the chromosome, in the DNA chromosomes sort of same, right? In the DNA, that codes for a protein. That's what you call as a gene. So the answer should be a section or part or length, right? If you have written section, part or length of the DNA, or you can also write of the chromosome, right? Because DNA, chromosome, same thing. Its chromosome means DNA with histone proteins. That codes for a protein. If you have not written protein, if you have written polypeptide instead of protein, you can get that mark as well. So, but you can't write strand, section, or you can't write strand of DNA that codes for a protein. You must mention whether it's a section, a length, or a part, right? Then you can get this 1 mark. Then they have said give the possible genotypes of a chicken with normal feathers. Genotype means the genetic composition, right? So we now, in order to, that means we are the ones who have introduced this genotype so that we know what it is. So genotype is basically written to show what their genetic composition is. So if a chicken is going to have normal feathers, right? That means, you know, the allele for normal feathers is capital F. Usually a capital letter is written to indicate that it is the dominant feature or the dominant allele, right? There is a dominant allele and there is a recessive allele. So dominant allele is always written in capital in normal genetics, right? When we are doing genetics, capital letters are used to indicate the dominant features. So capital F is for the normal feather. So if a chicken is going to have normal feathers, you know, a genotype is composed of 2 alleles, right? The genotypes are composed of 2 alleles like this: capital F, capital F, capital F, simple F, simple F, simple F. These are the possible genotypes of the chickens for the feather structure. Out of these genotypes, right, what is the genotype which codes for normal feathers? To code for normal feathers, right, to code for normal feathers, it should have the dominant, right? The dominant allele in it, because the dominant allele, that means it's actually dominant over the recessive allele. It covers up it shadows the recessive allele. That means when the dominant one is present, you can't see the recessive one, right? He's like a leader. When the leader is there, you can't see the team members, right? That means a dominant person is there. You don't care about the other people. So capital left, both are dominant. Both can be seen very clearly. So they will give the normal feather. Capital left, simple left, still there is a dominant one. So you can see the dominant feature, which is the normal feather. But when simple left is there, nothing is dominant. Both are recessive. Here only you have the silky feather chicken, right? So normal and normal, capital left, or capital left, simple left. So the possible genotypes you have to write, you can't write the word O, you have to write the word and, right? The word and should be there in your answer. Capital left, or else you can put a comma. Capital left, simple left. You can put like this, or else you can put simple left, capital left also, okay? The order doesn't matter. Both will create the normal gene, right? Then you had write either and, or you have to mention comma. You can't write O, okay? Because they are asking to give the possible genotypes. My answer is wrong. You had to write and or comma, right? Then a scientist investigates the inheritance of feather types in a chicken. The diagram shows a family pedigree for some chickens, okay? Right. How many chickens in the family pedigree? Pedigree are heterozygous. Right. Now you can see the black ones indicate we don't have to care about the male or females at the moment because they have not asked about male or females. So basically, the black ones indicate they are having silky feathers. Silky feathers means what's their genotype? Definitely all of them are having simple left, like this, right? Okay, this one is having simple left, and this is a parent, right? These are the parents, these are their children, not this one. This is an outside person who's marrying the sixth one, right? So we can't consider this as a person, this as a child. So you can see the one of the children is also having a recessive one. That means one of the children is also having silky feathers. For the child to have a silky feather, you know, from the mother's gene and from the father's gene only, the children genes are made. So one particular allele should come from the mother, one particular allele should come from the father to contribute to the children's gene, to contribute to the children's genotype. Definitely, this father should contain a capital left. Why is that? Because he is having normal feathers. But we have an issue. Is it capital left, or is it capital left, simple left? Right, that is the issue that we have. Right, it's easy to solve. You know, this is simple left. For this first child, this is the first child in a pedigree chart. I think it's not in your syllabus, but it's okay. I have to double check whether it's in your syllabus or not, but I think it's not. In a pedigree chart, always the first one in the line is the first child, but you don't have to know about that, right? It's just the first one in the chart is the first child, but you don't have to know much about that because it doesn't play a big role in the pedigree chart, right? So the first child, second child, it doesn't matter for us. So you can say capital left, simple left is there. So you can say simple left child is there. To create a simple left child, the father should contain a simple left allele. Otherwise, how can if a mother is giving a simple left allele, the father also should give somehow a simple left allele. That's how the child should have both simple left and simple left, right? Therefore, the father is not capital left. Father is capital left, simple left. So they're asking heterozygous. What do you mean by heterozygous? They are not having the same allele. They are having two different alleles. Capital F is homozygous, same allele. Simple F is homozygous, same allele. But all the others are, that means capital F, simple F is heterozygous, different alleles. So, what are the homozygous alleles? Easily, you know, the black ones are homozygous. Why is that? Because they are simple F. So they are homozygous. But if you take capital F, simple F, right? If you take capital F, simple, if you take the father is heterozygous. And if you take the children, you can't exactly, no, you can exactly say, you can exactly say because they are having normal feathers, right? Because they are having normal feathers, they have got the capital F allele from the father and a simple F allele from the mother. So definitely they all are going to have a structure like this. Capital F, simple F, capital F, simple F, right? So all of them are heterozygous. So how many heterozygous chicken are there in this pedigree chart? There are 4 heterozygous chicken. Yes, 4. Therefore, the answer is C. Then they are asking, use the genetic diagram to determine the probability of one offspring of individual 6 and 7 being a chicken with silky feathers. Okay, now 6 is going to marry the 7 one, and you can see they are going to have children, right? When they are having children, we have to cross their genotypes. So to cross this, you know, this the 6th one genotype is capital F, simple F. 7th one genotype is simple F, simple F. Yeah, so you have to cross them. You can easily do the cross like this. You can draw a table. I'll erase this table. I'll draw a new table. So you can draw a table like this, right? So in you can put mother over here. You can put father over here. So in this section, I'm going to write the mother's allele. Mother's allele is simple left, and father's allele is capital left, simple left. Now you can find the probable genotypes, the possible genotypes. Capital left, simple left can be there in their offsprings. Capital left, simple left again can be there in their offsprings. Simple left can be there in their offsprings. Simple left can be there in their offsprings. So they are asking, what is the probability of having a chicken with silky feathers? To have a chicken with silky feathers, they should have simple left. Out of the 4 possible answers that we are getting, how many genotypes are having silky feathers? 2 genotypes. So 2 out of 4 possible values, we are getting silky feathers. So you can convert it into a percentage. How much? 50% or half. The probability is half or 50%, or you can write 0.5. Either you can write it as a percentage, you can write it as a decimal, you can write it as a fraction. You get 4 marks for that means for mentioning that parental genotypes. Yeah, for mentioning the that means if you have drawn the Punnett square like this, you can get 3 marks easily. Otherwise, if you are not drawing the Punnett square, you have to mention the workings. Otherwise, you don't get the full marks. Right now, you could see in the previous workings. Yeah, in the previous questions where we were supposed to do the workings, we did not get marks for the workings. We directly got the full marks for our answer. But here, no, you have to write the workings to get the full marks. So for the final answer, you get just 1 mark. For drawing a Punnett square like this, you can get 3 marks. But when you're drawing the Punnett square, it should be complete. For example, it should have father, mother, or you have to draw the symbols. This is father, this is something, this is mother, right? I don't think those symbols are there. Those are like the standard symbols. So you can write father and you can write mother, or you can write female, you can write male, right? Something like that, right? And when you're indicating F and M like this, right, better to mention what it is, because the examiner don't know, right? When you are saying F, better to mention that it is the father, better to mention that it is the mother. So the the examiner will understand. Always give keys so the examiner can understand. Otherwise, this is the way that you can write the answer. You can write parental genotypes. Are capital F, capital F and simple F, simple F. For recognizing that the parental genotypes are capital F and capital F, simple F, and simple F, you get 1 mark. Then you can write down what are their gametes. Gametes means they are eggs, right? From the simple F, from the mother you get the simple F gamete. From the father you can get the capital F gamete. You can get the simple F gamete. That means the alleles are separated. See, I separate the alleles and I take them into gametes. Because when you're making the gametes, there is a reduction. Half of the alleles are present in the gamete. Because gametes are produced by meiosis. Meiosis means 50% is there, right? That means meiosis means, now if you take a diploid cell, it's two-way. When it is producing an egg, or when it is producing a sperm, it becomes, egg or sperm means gamete. It becomes N, right? It is getting reduced. Likewise, when they are producing gametes, you just have to write 1, capital F, simple F. They are getting divided like this. And since this is simple F, since both of these are same, it creates gametes of simple F, right? The father could create sperms having capital F allele, simple F allele. Some sperms will contain capital F allele. Some sperms will contain capital F allele, right? So you have to mention that also. Gametes are F, capital F plus F, and simple F, right? Then you have to write down the F1 genotypes. That means the possible genotypes that you can get in the F1 generation. F1 generation, that means the first generation of offsprings. That means the first generation of offsprings. That means this is the parental generation. Okay, I'll draw it, right? Otherwise it's complex. I'll draw it quickly. Parental genotype, right? So parental genotype is capital F, simple F, simple F, simple F, capital F, simple F. Then you draw the gametes, right? Better to write down in full words, gametes. You have to draw capital F gamete, simple F gamete. This is simple F gamete, see? You can draw arrows if you like. Then you can draw the F1 generation. Yes, F1 generation. Then you can write down the possible, you can write down the possible F1 generation. Capital F, simple F. Simple F, simple F. These are the possible F1 generation. 1 mark, 1 mark, 1 mark. Final answer, 1 mark. That's how you get the 4 marks, right? Okay. Did you understand that part?
Yes, miss.
Okay. The third question, the scientist observed that chickens have either normal feathers or silky feathers. However, the chickens have a wide range of different heights. Explain why there is a wider range of variation in height than in feather type. Okay, so here the main thing is, for feathers, you don't have much of a that means if you take feather, it's just either this type of feather or either that type of feather. But if you take height, it could vary, right? It has sort of like a shape like this, right? A bell-shaped curve, right? So the height is actually continuous, but the feather is not but the feather is not continuous. It is discontinuous. So if you mention this point, you get 1 mark for that. Not just that, you know, height depends on different genes. Different genes contribute to the height, right? That means in our chromosomes, there are several genes. All of them combine and contribute to our height, not just one particular gene. So height is polygenic. And in other words, you can write height depends on combination of many genes, right? In other words, polygenic means height depends on combination of many genes. Not just that, height depends on our environmental factors as well, right? If you are not taking enough proteins, no matter, even if our parents are high, even if our parents are tall, just because our genes say that we are getting tall, if we did not take enough proteins during our growth spurt, we'll never grow. We'll never increase our height. So height depend on our environmental effects. And also, you can, if you have mentioned about the height in that way, if you want to explain about the feathers, you can say feather structure happens only due to one gene because it is monogenic. You can mention like that. And there is another thing that you can explain, that the height depends on sex as well. That means sometimes usually males are usually taller than the females. So but feather type, it doesn't depend on the sex like that. So height, the genes of height, there are on our sex chromosome. You know, X and Y are the sex chromosomes in a male. X and X are the sex chromosomes in a female. So sometimes our X chromosomes can have this height, can have the gene for the height. So sometimes Y chromosomes can also have the gene for the height, right? So sometimes people having the height, the people having this Y chromosomes, that means males, they can become taller than us. They can become taller than the females. But there are that means the genes that control height, that means the genes that control the height are located on several DNA, not in just one DNA. Right? That's why we say they are polygenic. So if you have mentioned any of the three answers, you can get the 3 marks. Right. Now, here they're asking, the diagram shows how the alimentary canal with structures labeled X, Y, and Z. Okay, X, Y, and Z. I'm covering this part, right? X, Y, and Z. So they're asking which of these structures produce amylase. So X is basically, they have labeled the salivary glands over here. X is the salivary gland, and you can see Y is the stomach and Z is the pancreas. So you know our salivary glands, they produce some enzymes which helps in the initial digestion of carbohydrates in our mouth. What is that enzyme? You know, the digestion of proteins, lipids, they do not start in our mouth. They usually start in our stomach or small intestine, depending on the substrate. But carbohydrate digestion starts in the mouth. Why? Because amylase is there. Salivary amylase is present. So amylase get digested, carbohydrates gets initially digested in our mouth using salivary amylase. But if you take Y, which is the stomach, it has pepsin, right? It has the enzyme pepsin. What does enzyme pepsin do? It catalyzes the breakdown of proteins, not the breakdown of carbohydrates, not the breakdown of fats. So Y contributes to the breakdown of protein. So they do not produce any amylase. But if you take Z, pancreas produces the enzymes required for digestion of almost all the molecules, almost all the, even proteins, fats, carbohydrates, almost all these molecules. And they release these enzymes into our small intestine. So therefore, Z also produce amylase. X and Z are the structures. The pancreas and the mouth salivary glands are the structures which create amylase. Okay, so here they have given what amylase do. You can see amylase converts starch into glucose, and protease converts proteins into amino acids, and lipases converts lipids into fatty acids and glycerol. Basically, glucose, amino acids, fatty acids, and glycerol, they are the subunits, right? They're the building blocks of each of these big structures that they have given. Glucose is the building block of starch. Amino acid is the building block of proteins. Fatty acids and glycerol are the building blocks of lipids. Right. Here they have given table 1 gives the name of some enzymes. They digest and the okay, start by amylase or maltase, not into glucose. It should be maltase according to your syllabus. Maltose according to your syllabus, right? It should be maltose as they have given, right? So you can see table 1 gives some of the enzymes that the molecules digest and the product form. You can see the amylase enzyme which creates maltose is amylase, or you can write down maltase as well. It breaks down starch into maltose. And lipid is the molecule. Lipases are the enzymes which converts lipids into fatty acids and glycerol. But I took this from an IGCSE Edexcel recommended book. So maybe this part they have made a bit of a mistake over here. Doesn't matter, right? You can see proteases converts proteins into amino acids, but they have given marks for polypeptides as well. Yes, they have given marks for polypeptides as well. Polypeptides or peptides. You can write any of those. Right. And here it doesn't have to be fatty acids and glycerol. It could be fatty acids or glycerol. But I believe since lipase breaks down lipids into their subunits are both fatty acids and glycerol, right? Fatty acids and glycerol. So there's no harm in writing fatty acids and glycerol. But since they're asking just one product, if you want, you can write down fatty acids only, right? Now, they have not, that means they have not deducted marks for writing both fatty acids and glycerol, because that is the most appropriate answer. Okay, here they have given table 2 shows the recommended daily amounts of some dietary components of a person. In the last paper, in the very first paper that we did also, RDA values were given, right? Here also they are giving the RDA values. So usually they ask the animal nutrition part in terms of these RDA values by giving tables and everything. That's how they check your knowledge on the animal nutrition part in your syllabus. Right. You can see 100 grams servings of lentils provide 25 grams of proteins. Okay, 100 grams of lentils provide 25 grams of proteins. Then they're asking to calculate the mass of lentils that contain 46 grams. This is an easy calculation. If 20, that means you have to divide 100 by 25 to get the amount of lentils to provide 1 gram of proteins. Then you can multiply it by 46 grams. So 100 divided by 25, you get 4. That means 4 grams of lentils provide 1 gram of protein. That means 46 grams of proteins. To provide 46 grams of proteins, you have to multiply 46 into 4. You get 184 grams. Remember, that means here they have given the grams, but if they have not given the grams, you must mention grams. Right. Discuss the possible long-term effects of this person eating the same diet every day. Okay, this is the recommended daily value, getting 2200 kilojoules. But look at this. Here you see they have used the word discuss. So you are allowed to write your opinions in this, right? You can see RDA value, the recommended daily value is 2200. But the diet of the value that he is taking in consists of 2700 kilojoules. So what happens? He's taking in excess energy. You can write down that particular person is taking in excess energy. And you can write your opinion. It will lead to obesity, right? Taking in excess energy, lead to obesity. Or else you can write down taking in excess energy will increase the risk of having diabetes, right? If you have written any of these, you get 1 mark. This is for this 1 mark, for this 1 mark. Then compare the proteins. Proteins, you can see 46. He's having the correct amount of proteins. So you are discussing it, right? So write that down as well. He's taking enough protein. So write your opinion. So if he's taking enough protein, the growth should be normal. See the words, these discuss, explain, describe, these words are really important in your question paper. So you can see enough protein, so growth should be normal. And the vitamin A levels are sort of high, but actually it's not high by a great amount. Now, if you compare 2200, 2700, there is a big difference. But here that's not much of a big difference over here. So you can just consider it as the same. So enough vitamin A, so no risk of night blindness. And also the diet of this person, it's just in 1 day, right? So now here, even though they have said he's eating the same diet every day, sometimes the vitamin A, that means even though it's just an increase of 0.02, which cannot be considered actually. That is why they have taken it as, that's why they have considered it as enough. So don't consider it as a high level, right? If it is something like 0.5 increment, then you can consider, because vitamin A is considered in milligrams. So milligrams, even 0.5 milligram, it's a high value, but 0.02, it's a negligible value. And 65 milligrams in vitamin C, 32 vitamin C levels are less. So you can say the vitamin C levels are less and it would lead to scurvy, right? Because the deficiency of not having vitamin C is leading to scurvy. And calcium levels of that person are also less. So if that person is taking calcium like this for a certain for a certain amount of days, that will lead to osteoarthritis or what? Or rickets. If that is a child, it will lead to rickets. And iron levels are almost the same, right? Just one. So no issues, right? Yeah, enough iron, so no risk of anemia. And also fiber, it's a very low value, not just 1 or 2, almost like 14, right? Around like 14, that means when compared to the RDA value, 14 grams of fibers are less in that diet of that person. So 5 is less, so there is a high risk of constipation, not low risk. High risk of constipation is there. High risk of constipation is there. High risk, right? So you get 5 marks for writing that answer. Yes. Then they are asking suggest 2 reasons why the RDA may not be the actual amount required by the person. Even though they have mentioned that this RDA is the required amount, sometimes you need more than that. Why is that? Because then, even though they have said like that, different people are different from each other, right? People are different. This is just for a typical average person that they have considered. But sometimes it could be that means for females it could be a different RDA value. So depending on the gender or the sex, the RDA value can change. And depending on a person's body mass, if a person is bigger than the usual level, right, that person's body mass is high, so that person requires more energy for his or her day-to-day activities. And even though this RDA value is the required value, sometimes on that particular day that person might be doing a lot of extra work, right? Working harder than the usual levels. Yeah, so therefore the activity, the exercise, the lifestyles also affects. And for a pregnancy woman, the values are totally different. It needs almost like twice the RDA value that they have mentioned. And also some people have different metabolic rates. No matter how much you are eating, sometimes you can be thin. That's because you have an increased metabolic rate in your body. So different metabolic rate people require different amounts of values. And also age. Sometimes for small people, it's not the required amount that they need. For growing people, they need sometimes more than this. So it could change, right? So if you have suggested any of the answers that they have given, any of the 2 from here, you get 1 mark. You get 2 marks. Then they have mentioned the skin is an organ involved in temperature regulation. They have asked why skin is described an organ. So you know our basic component of living organism is the cell. Then we have the tissues, because different cells they combine and they make the tissues. And then tissues combine and then they make the organs. And all these organs combine and they make the organ systems. Finally, different organ systems combine: circulatory, nervous, excretory, respiratory, all this combine and make the full organism. So why is skin described as an organ? Because skin has several groups of tissues, right? It has a epithelium, it has a dermis like this, epidermis, dermis, it has a subcutaneous layer like this. There are several layers like that. Because of that, and also there are several blood vessels, right? Because of that, it's made up of several groups of tissues. Organ means it's made up of several groups of tissues. So skin is described as an organ because it is made up of several groups of tissues. So for that, for several or different groups of tissues, you get 1 mark. And also different cell types made up of different cell types. This time they have allowed that mark. But better not to include the word cell over here, because organ means made up of different tissues. Here they have given mark for including made up of different cell types, but they might not always give that mark. And also, if you have just written made up of tissues, instead of writing made up of different groups of tissues or made up of several groups of tissues, no marks for that, right? So you must mention several or different. Then only that
I
one
think
mark
I've
is.
just written I've just written
Yes.
this because the skin is made up of multiple tissues, so I don't think I'm getting that mark.
Multiple tissues. I don't think you will get that mark, dear, because multiple tissues, several groups of tissues, multiple tissues. No, because multiple could be the same. That means okay, multiple is not the same type. Multiple means different types, right? Okay, multiple means different types. Multiple tissues. Multiple tissues means, I think you will get that mark, because multiple means, now multiple doesn't have the idea of the same tissue, right? Multiple means a variety, right? Sort of like different, right? So you have that point in your answer. Multiple, the word is there, but don't make it too complex next time, right? Just write the word different. Okay?
Yeah.
But because you have the word multiple has that meaning, you can get that mark, right? Then the B1, the diagram shows a section of human skin. Explain how the skin capillaries regulate human body when the body temperature increases. You know, these are the capillaries which are there. When our body temperature increases, our blood vessels expand. We don't say it expands, we say it dilates, right? So our blood vessels dilates, and then more blood flow through our blood capillaries. When more blood flow, you know, blood is the liquid which carries the heat around our body. So when more blood travels through this blood capillaries, you know, the blood capillaries are closer to the skin. When they get expanded like this, they are even closer to the skin. So what happens? The heat gets radiated to the outside of the body quickly. That's why more blood flows. Previously, it couldn't happen. Why is that? Because the blood capillaries are thin, only less blood flow, and only a small amount of heat was radiated. But now, since the blood capillaries have dilated, the process is called vasodilation, and that happens due to homeostasis. That means blood temperature. That means homeostasis could be either regulation of regulation of body temperature, regulation of blood glucose level. That means we have a certain body temperature, 37 degrees Celsius, which is the typical. Well, it could be from 36.5 degrees Celsius to 37.5 degrees Celsius. So 37 degrees Celsius is the optimum temperature in our human body. So if it increases beyond that, our hypothalamus, right? Our hypothalamus sends signals to our hypothalamus is the thermostat. Thermostat means in an iron also there is a thermostat, right? If you take an iron, if you have plugged the iron for a long time, it makes a tick sound and that light switches off, but you are still plugging it. Why is that? Because the temperature of the iron has increased above a certain value, and the thermostat in the iron is has detected that, and it has quickly disconnected the ability of the iron to get heated up further. And after it has cooled down, again the thermostat recognizes it. Okay, the iron has reached a normal level now. Now I should increase the temperature back again. Again, that light will switch on. That's why when you keep the iron plugged in for a long time, when you're ironing clothes, the light goes off and the light comes again after a certain time. So likewise, in our body also there is a thermostat which is called the hypothalamus. The hypothalamus will detect the temperature increase, and it will send signals to our blood capillaries. So our blood capillaries will dilate. And when they dilate, more blood will flow. Not just that, even our sweat glands, right? Our sweat glands secrete more sweat, because when more sweat gets secreted, sweat gets deposited, and when the sweat means water with salts, right? So when water gets excreted onto our surface like this, water absorbs the heat from our skin and gets evaporated. Because to get evaporated, they need heat. They absorb the heat from our skin surface. That's why we feel cool when there is sweat. After the sweat has evaporated, we feel cool. So that's a body mechanism to cool our body. And also when we get out from a swimming pool, we can see when the water on our skin surface gets evaporated, we feel really cool, right? And the same thing happens with the sweat. So basically here you have to write, they're asking how skin capillaries regulate the body temperature. You can first write vasodilation occurs. As a result, there will be increased blood flow near the skin, or increased blood flow to the skin, or increased blood flow through the capillaries. And also you can explain what vasodilation is. Vasodilation means the expansion or the widening of the capillaries or the blood vessels, right? The expansion or the widening of the arterioles or the blood vessels, right? But you can't write capillaries widen. They are not giving marks for capillaries widen. You can write vasodilation of capillaries. Capillaries dilate, right? Capillaries dilate. You can't use the word capillaries widen. You had to write capillaries dilate. Capillaries dilate, but arterioles, they have used the word widen. You can use arterioles widen, but for any blood vessel, what I believe is it's not safe to use the word widen or expand. Because in blood vessels, you use the word dilate and constrict, not contracted, constrict, right? To tell that the blood vessel has been smaller, that the blood vessel has got smaller, you use the word constrict. That happens in vasoconstriction, right? So I think in your paper, it's always safe to write dilation and constriction, these words. Right, so 3 marks for these 3 points. Now let's get into the next question. Yeah, we have a lot of questions. Let's quickly do. Yeah. Here they have asked, some people have suggested that drinking warm drinks may cool the body because it increases the rate of sweat production. They have asked us to include the experimental details in your answer and write in full sentences to design an investigation to determine whether when we drink warm water, whether it increases our rate of sweat production, right? So we are going to get people, take some people, we are going to give them some we are going to give them some warm water, and we are going to check whether the sweat production increases when they drink the warm water, right? We know it happens, but we are going to do an investigation to see whether it really happens or not. So when you are writing an experiment, you always have to make sure that the experiment that you are doing is fair. To make sure that the experiment that you are doing is fair, all the variables that you are taking in should be of the same level, or should contain similar features. So if you are taking people, you must mention that all these people are either of the same age, or of a similar category, or either they have the same mass, or either they are of the same gender, or they have the same fitness, or their body temperature is equal, right? So you must mention that first, and then you can't just write like this, right? You have to explain the experimental details. You have to give all of them the same amount or the same volume of warm drinks, right? First, that means here, same exercise, same food, water, volume drink, this should come second. Even though the marking scheme has said it in this order, you must mention it in the correct order. That means that's how it makes sense, right? So you give them the same volume of drink, right? Same volume of drink should be given to all of them. And then you have to give the same that means you have to give each of them a cotton wool, because you can't write weigh the cotton wool before writing. Then if you didn't write that, you're giving them a cotton wool at first, right? So you have to describe it. You're giving them a cotton wool to measure the amount of sweat that they are excreting from their body. You're placing a cotton wool in their arms, right? Or something like that, right? You have to mention it in your own words. And then you measure the mass of sweat, right? You weigh the cotton wool, so you can measure the mass of sweat. That means when you're giving a cotton wool also, it's better to say that you are giving the same mass of cotton wool to everyone. And then finally, when you're calculating the final mass, you can get the difference. By calculating the difference, you can measure the mass of sweat. Otherwise, you can't measure the mass of sweat like that, right? So you can give more, you can explain, you can give more experimental details, but they are giving marks for these ones, right? That means even here, they have not asked us to explain it. But it's your responsibility to make some sense and write this, because you can't write the answers like in the marking scheme. Even though they have asked us to include experimental details, better to explain it and write it, including this point, right? To make it more sensible, right? So if you have written any, did you understand this part? You have to use the
Yes,
same
yes.
temperature, yeah, and you can repeat the experiment to get the average result. That means to increase the reliability of the result, you can repeat the experiment with different ages. That means rather than taking the same group of people, you can take different groups of people and you can calculate you can take the average. So you get 6 marks for that. Now the 8th question. Right. Since they're asking 2 abiotic variables that the student controls, that means here the student uses a method to investigate the effect of fertilizer on the growth of plant seedlings. So here they have given how they are doing it, how the student is doing it, and they are asking us to mention 2 abiotic variables that the student controls. So when you are planting a seedling and when you're allowing it to grow it, you know this seedling requires sunlight. This seedling requires sunlight. Not just that, the seedling also requires water. It requires water for it to germinate. And also this seedling requires fertilizers or compost. Why is that? It needs nutrition for it to grow. So even though they have not mentioned over here, you are the one who should know for the growth of a plant that they require these things. So with that knowledge only, you can say light intensity is given. What light intensity is a abiotic variable that the student can control. The amount of water, don't write amount always. In this Cambridge and Edexcel examinations, you always have to give the particular that means the measurement should be either volume, mass, something like that. When you write amount, it's not science. To make it like in science papers, what these Edexcel Cambridge boards are expecting you is you are writing it in the scientific way. So always make sure you write water volume, right, rather than amount of water volume. And also mass of compost, rather than writing amount of compost, mass of compost. If you have given any 3 examples, any 2 examples from this, you get 2 marks. And here they're asking, the student dries the seedlings in an oven to find their dry mass. Suggest why is it important to use dry mass in this investigation. So, actually, the real biomass. Now, there is something called biomass in all the organisms. Biomass, we consider biomass, the real biomass is the biomass without water. If you take an organism, the biomass of that organism is the mass of that organism without the water content in its body. So biomass is the dry mass of an organism, right? Biomass is the dry mass. So what is important is the dry mass, because if you consider the wet mass of an organism, different people will have different water levels in their body. So likewise, different seeds will have different water in their body, different water in that means in themselves. So that could differ the results. Different water content means you can't have a fair result, right? Some seeds could absorb more water, so they could have a higher mass, but that's not their real mass. That's their mass with water. So in order to compare their real mass, you must take the dry mass. Otherwise, that the result will not be accurate. Therefore, the water, you must mention that water is not a biomass. Dry mass is the true biomass. If you have mentioned this point, you get 1 mark. And also water content. When there is water, it increases the mass. So different seeds will have, if you have given this idea that different seeds will have different, that some seeds will have more water than the others, you can get 1 mark for that. And also if you have mentioned that water content increases the mass, there also you get 1 mark, right? And also taking the dry mass, it makes the comparison valid, right? Comparison valid means dry mass means everyone's mass is going to be, that means the real mass without water. So it will be a fair what do you call, experiment. Therefore the comparison is valid. For that also you get 1 mark. Then they have asked us to plot a line graph to show the dry mass of seedlings without fertilizer and the dry mass of seedlings with fertilizer from day 4 to day 20. Okay, so here, you have to at least use half of the grid. If you have just drawn a scale like this and if you have drawn the 2 lines like this, you won't get any marks for that. You have to use at least half of the scale, and make sure you always label the 2 axes, right? You must measure the two axes. One should be the dry mass of seedlings, that should be in grams, and the other should be days, right? And then you can draw it like this: 0, 1, 0, I'll divide it like this: 1, 2, 3, 4, 5, 6, 7, 8, 9, likewise. And here you can mention the days like 2, 2, 4, 6, 8, 10, 12, 14, likewise finally 20. Okay, in terms of 2, 4, 6, 8, 12, 14, 16, 18, 20. Did you get that? 2, 4, 6, 8, 10, 12, 14, 16, 18, finally 20 over here, right? So on the 4th day you can see, so you are using most of that means more than half of the grid. So that point you will definitely get. Then you have to correctly label the values. If you have drawn a bar chart, you won't get any marks. This should be a line graph. When they say line graph, they are not asking you to draw the best fit line or anything like that. They're asking you to join the lines, right? Join the lines. It could be anything. You have to join the lines. So on the 4th day, without fertilizer, and also when you have labeled it without fertilizer and with fertilizer, you have to mention what is without fertilizer and what is with fertilizer as well, right? That means what is the one which is without fertilizer and what is the one which is with fertilizer, right? That should also be mentioned. But that won't get any, but you won't, that in the marking scheme this time they have not given marks for that. That means mentioning that part, they have not given marks for that. But in order to like be more accurate, you can label the that means you can label the 2 graphs, because you are drawing 2 graphs, right? So you have to mention what it is, with whether what is the one without fertilizer and what is the one with fertilizer. So 4th one, you can see 4th one is 3.5. So here is the 4th one. It's 3.5. Somewhere over here it starts from here. So without fertilizer it starts with 3.5. With fertilizer it starts with 3.8. So it starts with 3.8 over here. Shall I send you a picture of this what do you call this graph, so you can mark your graph later?
Yes, miss. Yeah, that'd be helpful.
Okay, I'll send you a picture of this graph to the WhatsApp. And yesterday also I said I'll send some points as well. I'll send those as well today. Right. Now let's get into the next part. So altogether you get the 5 marks like this. I must mention that. For drawing the axis you get 1 mark. For drawing the axis correctly, you get 1 mark. And for occupying more than half of the space, you get 1 mark. So 2 marks for that. And also for using ruled straight lines, right? For using ruled straight lines and for joining the points, you get another mark. So altogether, you get 4 marks for that. And if you have mentioned all the points correctly, that means if you have just made at least a single mistake, no marks. If you have marked all the points correctly, you get 1 mark. And also you have to give the key. Okay, they have given the key. That means you have to mention what is what. What is this line and what is that line. You have to mention this is the line without fertilizer. For that also you get 1 mark, right? So 5 marks are like that for those. Yes, that's what I was also wondering, because in the marking scheme, in my first glance, I didn't see that. But I was wondering why they didn't give that. Otherwise, how could you find the 2 lines, right? So it's always better to mention what is the one without fertilizer and what is the one with fertilizer. Right. Then the next one, the fertilizer contains magnesium ions and nitrate ions. Explain the effect of these 2 ions on the growth of seedlings. Okay, so yesterday we also learned in plant nutrition, they need magnesium ions and nitrate ions. Why do they need magnesium? Yeah, why do they need magnesium ions? To make chlorophyll. To make chlorophyll or chloroplast, right? To make chlorophyll in the chloroplast, or to make chloroplast also, okay, right? So here they're asking the explain the effect. They're not asking what is the purpose of chloro. They're not just directly asking us question. They are expecting us to explain why do we need chlorophyll, right? Why do the growth of seedlings need magnesium? To make chlorophyll. Yes, for what? That means so more photosynthesis can happen, right? When you have more chloroplast, more photosynthesis can happen. And also you can further explain it if you like. So to make chlorophyll, get 1 marks. And for more photosynthesis, you get 1 mark. To produce more carbohydrate, you can get another mark. To produce more carbohydrate, you can get another mark. So altogether, you have 3 marks, right? You have altogether 3 marks over here. Then you can mention about nitrate ions, right? Nitrate ions. Why do we need nitrate ions? For amino acids. We need nitrate, to make amino acids. And amino acids for what? Amino acids for the growth of the seedlings, right? For the synthesis of, that means basically for the growth of seedlings. So for this you get 1 mark, since they are important in the growth of seedlings also. If you have written that, you get 1 mark. But rather than that point, for magnesium, you have several important points where you can get the 3 marks, right? But it's important to write the nitrate one as well. So altogether, you have 4 points for that. Then in the 9th question, they have asked us to, they have given a pea flower with structures labeled X and Y. They have asked us to mention the name of X and Y. You can see X is just a single structure which is protruding like this, and it is the stigma, because you can see it has an area where the pollen can actually get placed on. Therefore, it's the stigma. And you can see these are the these are the anthers. These are the stamens. You can see the filaments are there, and also they have the anthers where the pollen is produced. Therefore, X is the stigma, Y is the anther. Then they are asking, explain how the 2 structures present in the diagram show how the flower is pollinated. That means how stigma and anther shows us how this flower is pollinated. Okay, by looking at the anther and the stigma, you can see both anther and stigma are inside of the flower. They are not outside. Usually, anthers and stigmas are outside if it's a wind pollinated flower, because then the anther could be easily carried away, then the pollen in the anther could be easily carried away by the wind. But this time the anther and the stigma are inside. That indicates it's a what? It's a insect pollinated flower. So how is the flower pollinated? First mention that it's pollinated by insect pollination. Then how do we, before writing if you want, you can write this as well because it has large petals, right? But here they are asking, explain how the two structures, the two structures that you have mentioned, right? How those two structures show that the flower is pollinated. So anthers are not hanging out. In a wind pollinated flower, anthers are hanging out. But in an insect pollinated flower, no, anthers are not hanging out. They are almost the same size as stigma. So when insects come, they brush against the anther and they deposit the pollen onto the stigma. That's how self-pollination happens. So stigma is also within the flower. Because of these two reasons, we can conclude that this flower is pollinated using insects. Right. Then the next part, let's quickly do this part. State which carbohydrates the scientist identified in the ungerminated seeds and the germinating seeds. You can see the ungerminated seeds. Yes, ungerminated seeds. The color of the iodine solution is black. Right. If black present means that means the color of iodine solution turns into black if starch is present, right? So starch is present in both germinated as well as ungerminated seeds. But if you take the glucose, or if you take the sugars, right? If sugars are present, the Benedict solution will turn red. So you can see sugars are only present in the germinating seeds. So ungerminated seeds, what is the carbohydrate that they have? They have starch. But if you take the germinating seeds, the starch of them is being converted into sugars. Why is that? Starch is the storage polysaccharide, or starch is the storage carbohydrate. You can't use starch for the growth and all the other purposes. You have to convert, or you have to break down the starch into smaller units like glucose, maltose, right? Or sucrose, and these are being used in the growth purposes. But why can't you write sucrose over here? Because sucrose won't convert Benedict solutions into red. Sucrose can't do that, right? Sucrose is not a reduced in sugar. Glucose and maltoses are reduced in sugars. Germinating seeds have sucrose as well. But more than that, the reason why the color changes is because it has glucose. Right. Then they are asking, explain the difference in the carbohydrate composition of the germinated seeds and the ungerminated seeds. Right. You can say like this: in germinated seeds, starch is used for storage. Right. Starch is used for energy storage. Right. And why is starch used as storage substance? Because starch is insoluble, right? And because starch is insoluble in water, it doesn't get involved in other processes. So it does not affect osmosis. Because if starch is soluble, it will reduce the water concentration. So the water concentration in that particular plant will reduce, right? That means when a particular solute is present in water, we don't say that the water is diluted. We say water is concentrated. So starch won't affect osmosis since it's insoluble. So in germinated seeds, you can mention these two things about starch. Then ungerminated seeds, not in germinated, ungerminated. In germinated seeds, you can mention that water, now in germination, first water enters into the seed. When water enters, it will activate the enzymes present. So when water enters, you can mention water activates the amylase or the enzymes, right? Water activates the enzymes. When water activates these enzymes digest or break down the starch. Into maltose or glucose. That's why you have maltose and glucose in the germinating seeds, because when water enters, starch is being converted into maltose, and glucose. And you can mention this glucose is used in respiration, because they're asking what the reason reason for the difference in the carbohydrates between ungerminated and germinated seeds. The reason is because in ungerminated seeds, starch is stored as the energy store, and since it's insoluble, it does not affect osmosis as well. And also in germinated seeds, when water enters, it will activate the enzymes, and these enzymes can convert the starch into glucose, maltose, or sugar, and this glucose can be used for energy. So if you have written any of those points, you can get the 3 marks. Then they have asked, explain why jars are used in the investigation, why jars used in the investigation are not sealed, right? So here, the reason why they have not sealed is because now when seeds are germinating, they need oxygen as well, because the plant seedlings, they are carrying out respiration as well, right? That means these seedlings are carrying out respiration. Both photosynthesis, respiration takes place, but in a germinating seed, we mainly consider the respiration process. So basically, to allow this respiration, oxygen should enter into these cells, right? And carbon dioxide should escape. So because of this, we can't have the jar closed, right? So to allow the oxygen in and to allow the carbon dioxide escape, to allow the oxygen in for germination, that's highly important. And to allow the carbon dioxide escape, you get 2 marks. Right. I will be stopping the discussion from here. Next week, I will be, no, only a small part. No, a big part is there. I will be stopping the discussion from here. Next week, I'll take this part into consideration and I'll discuss the paper. Right? Okay.
Yes.
All right. So I hope that you have understood this part. If you have any doubts, you're free to text me anytime, right? Any questions? If you are doing any extra papers also, you can ask me any questions anytime. I'm open to questions. I'll be closing the discussion now. Study well, because you have around like 4 months. Yeah. In April, May, in May, June, right? You're doing?
Yeah, May, June.
Yeah, so you have around like 4 to 5 months left. So you need to work hard during this time and target for an A star, right? You have the knowledge with the answers and everything that you are giving. You can go for the band 9, right? That means the highest band. You can go for that. Study well, right? Good night. Okay.
Thank you, Miss. Good night.
Okay, welcome.